Let's start by understanding how our immune system is structured and how it functions. We have two main parts working together, the innate and adaptive immunity. Innate immunity includes cells like monocytes, natural killer cells, macrophages, and others that act quickly to recognize threats using signals like cytokines. Adaptive immunity involves B cells producing antibodies and T cells targeting specific invaders. Dendritic cells play a crucial role by presenting antigens to T cells, helping the system distinguish friend from foe. This collaboration is essential for effective defense. Now, focusing on the lymphatic system, it's key for lymphocyte growth and development. It's closely linked with blood vessels, allowing lymphocytes to circulate throughout the body. This network supports immune surveillance and helps cells reach sites where they're needed. When we talk about lymphoid organs, we split them into primary and secondary groups. Primary organs, like bone marrow and thymus, are where lymphocytes develop. Secondary organs, such as the spleen, lymph nodes, tonsils, and mucosa-associated tissues, are where immune responses are coordinated and executed. To get a better sense of where these lymphoid organs are, imagine a child's body. The thymus sits in the chest, the lymph nodes are scattered throughout, the red bone marrow is inside bones, and the spleen is also in the abdomen. Each has distinct structures, like the thymus's cortex and medulla, or the spleen's red and white pulp, which support their unique immune functions. Bone marrow is fascinating. It's not just a filler inside bones. It's the central hub where blood cells, including immune cells, are born. Stem cells here differentiate into various types, like natural killer cells, T and B lymphocytes, and others, that marrow environment with bone matrix and supportive cells nurtures this process, ensuring a steady supply of immune cells ready to defend the body. Bone marrow plays several critical roles in immunity. It's the primary site where blood cells form, including immune cells. It serves as a reservoir of stem cells that can develop into diverse immune cells. During infections, the bone marrow ramps up production to meet increased demand. Also, it supports the maturation of B cells, which are vital for producing a wide variety of antibodies. The thymus is an essential organ for T cell development. Located in the chest, it has distinct regions, the cortex and medulla, where immature T cells mature. The thymus contains specialized cells like epithelial cells, macrophages, and dendritic cells that help train T cells to recognize harmful invaders while avoiding attacking the body's own tissues. T cell development in the thymus is a carefully controlled process. Immature thymocytes undergo maturation and selection to ensure they can distinguish self from non-self. Most thymocytes don't make it past these checkpoints. They die off if they don't meet strict criteria. This ensures that only functional, self-tolerant T cells enter circulation, ready to protect the body. Lymph nodes act like checkpoints where lymph is filtered. Multiple vessels bring lymph into the node, where immune cells scrutinize it for pathogens. Macrophages and lymphocytes work together to detect and respond to threats. Lymph then exits through efferent vessels. Passing through several nodes helps ensure the lymph is thoroughly cleansed before returning to circulation. The spleen is the largest lymphoid organ and serves multiple immune functions. It filters blood, removing old red blood cells and capturing antigens. Phagocytic cells in the spleen defend against blood-borne pathogens. It also produces activated lymphocytes that enter the bloodstream, making it a crucial organ for blood filtration and antibody production. Immune cells can be grouped into three main types. Lymphocytes, including B cells, T cells, and natural killer cells, are key players in adaptive immunity. Phagocytes, like neutrophils and macrophages, engulf and destroy pathogens. Other cells, such as dendritic cells, Granulocytes and mast cells have diverse roles, from initiating immune responses to regulating inflammation. The immune system cells originate from hematopoietic stem cells that differentiate along two main lineages, lymphoid and myeloid. The lymphoid lineage gives rise to natural killer cells, T cells, and B cells, including their memory and plasma forms. The myeloid lineage produces neutrophils, eosinophils, basophils, monocytes that become macrophages, and dendritic cells. This differentiation supports a wide range of immune functions. Comparing phagocytic cells reveals distinct roles. Neutrophils primarily cause inflammation and quickly invade tissues, but have a short lifespan. Monocytes circulate in the blood as precursors. Once in tissues, they mature into macrophages, which live longer and present antigens to other immune cells, while secreting signaling molecules. Each cell type contributes uniquely to immune defense. Looking at dendritic cells, granulocytes, and mast cells, each has specialized functions. Dendritic cells, found in skin and mucosa, are crucial for presenting antigens to T cells and initiating immune responses. Granulocytes mainly reside in tissues and defend against parasites, releasing histamine. Mast cells, located in skin and connective tissues, also release histamine and play a key role in allergy development. B lymphocytes originate in bone marrow and are central to humoral immunity through antibody production. Each B cell has a unique receptor to bind specific antigens. They also act as antigen-presenting cells to T cells. B cells protect against viruses and bacteria and have the unique feature of not reacting to transplant or cancer cells, highlighting their precise targeting. T lymphocytes start in the bone marrow but mature in the thymus. They are essential for cell-mediated immunity. T helper cells activate other immune cells through cytokines. Cytotoxic T cells kill infected or cancerous cells, and regulatory T cells help keep immune responses in check to prevent overactivity. This diversity ensures a balanced and effective immune defense. Natural killer cells develop in bone marrow, but differ from T cells because they lack typical T cell receptors. They can kill infected or tumor cells without prior sensitization or antigen presentation. This ability places them at the intersection of innate and adaptive immunity, providing rapid responses to threats without needing specific activation.